Welcome to the series of organic chemistry. Dear students, we have been learning organic chemistry under Biomolecules chapter. Today we are going to learn about proteins. Under this, we are going to learn in detail about amino acids, their structures, and primary, secondary, tertiary, and quaternary structures of proteins, followed by enzymes, vitamins, and very important nucleic acids. Under this, we are going to learn about DNA and RNA, their structural elucidation, followed by hormones. Dear students, what are proteins? Proteins are the compounds which are fundamental structural units of an organism. It can be human or any bacteria, plants. All these are going to have proteins. These are the fundamental structural units. In Greek, proteo means the prime or important. That means these are the fundamental units of body or the matter. I can say fundamental units of living organisms. So these living organisms are going to have proteins. Then what are proteins? When you do the hydrolysis of these proteins, you are going to get amino acids. Amino acids. So what are amino acids? So the amino acids are the monomers of proteins. Proteins are the polymers of amino acids. So proteins, when they are having monomers of amino acids in a large length chain, are known as proteins, and the sequence of these proteins depends on the amino acid or the name of the proteins. Yes, let us see the amino acids. What are the common amino acids, or what are the naturally occurring amino acids? Yes, students. So proteins upon hydrolysis gives amino acids. In nature, there are 23 amino acids. In some textbooks, they have given only 20. Yes? So actually there are amino acids, are 23 amino acids. Then how to write amino acids? The amino acids are the compounds having, for example, this is the way, Fisher projection way. Amino acid, here R group, then it is H, and this side is amine group. Alpha amino acid. The amine group must be written towards left side. In a Fisher projection, highly oxidized group, has to be written at the top, then followed by next carbon and the remaining carbon chain. When you are coming from down, the amine which is having alpha to this carboxylic acid is towards left side in natural amino acids. Natural amino acids are having left side, means L amino acids. Dear students, please remember L amino acids. This is the configuration, L amino acids. And when it comes to S RS absolute configuration, these are S amino acids. This is S represents absolute configuration. How to reach this absolute configuration? For example, give the SIP rules. Based on SIP rules, give the priority numbering. Nitrogen is 1, carboxylic acid 2, and for example here it is methyl group, it is going to be 3. Now it is showing right clockwise direction, but the hydrogen must be come towards down. Then what to do? Do the mutual or two exchanges like this, like this. Then what you are going to get? You are going to get this amine goes here, and this carboxylic acid comes here, followed by hydrogen here, and this is methyl. And now give the numbering. What is the number? 1, 2, 3 and 4. So what is the direction? It is S, anti-clockwise direction. So alpha amino acids or natural amino acids are S configuration amino acids. Remember students, it is very important. All natural occurring amino acids are L configuration. Now this DL nomenclature is not going to be followed in modern textbooks. So it is absolute configuration is RS configuration, which follows SIP rules, that is Cahn-Ingold-Trillox rules. So based on that, it is S amino acid. Yes students. So this is how it is going to be S amino acid. Then is this a way to write on paper? No. So when you are going to write on paper, you have to write this is acid, carboxylic acid. For example, this is a methyl group. Write like this and put the amine group down. Amine group down. This is the usual way of writing amino acids. So when you are writing on a plain paper, write amine group down. If you write above, it is going to be different configuration. So follow this regular pattern for writing amino acid structures. Yes students, let us see the amino acids. So the first one is alanine. And before going into the structures, dear students, usually the amino acids can be represented by 3-letter abbreviation or a single letter abbreviation. Let us see alanine. Alanine is CH, CO2, H, and amine, and this is methyl group, and this is alanine. Alanine, and its abbreviation is ALA, or in a single letter I can write A. This is alanine, followed by second one is arginine. Arginine is having CH-NH2 amino acid. This is alpha amino acid. CH2, CH2, C1, 2, 3, 4. CH2 and guanidine having NH, C, double bond N, H and NH2. This is the guanidine group. This is guanidine group, arginine. The short name of arginine is ARG, and the single letter is, it is very important students, it is R. R. It is not going to based on the starting letter. Arginine is ARG. No. So alanine is A, arginine is R. Yes. So this is arginine, followed by asparagine. Asparagine is CH, CO2H, NH2, and this is CH2, CO, NH2, formamide of aniline, alanine. Dear students, we have been saying alpha amino acids. What is alpha amino acid? This is the position of alpha. So the carboxylic acid chain, for example, this, if I take this example, this chain is having alpha position is amine group and this is beta. If the amine is here, it is going to be beta amino acids, but naturally occurring amino acids are alpha amino acids, beta, alpha, beta, gamma, delta. So based on this position of amine, you can name it as alpha amino acid, beta amino acid, gamma amino acid, delta amino acids, but naturally occurring amino acids are alpha amino acids. So this is asparagine. So asparagine, this is basic unit of alanine, this is formamide group. Then what is the abbreviation of this? Asparagine can be written as ASN. ASN, and the single letter abbreviation is N. Then follows aspartic acid. And this is the simply you can write it as aspartic acid is alpha amino acid of succinic acid. Do you know the succinic acid formula? So this is succinic acid. Succinic acid is, this is succinic acid, right? 4 carbon dicarboxylic acid is succinic acid. And this alpha amino acid is aspartic acid. Its abbreviation is ASP and the single letter is D. Yes students, so there are 4 amino acids starts with letter A. Then there is no amino acid of B. Follows C, alphabetical order C amino acids. These are very very important students because these 2 amino acids are going to have sulfur. Sulfur is highly poisonous substance. So the 5th compound is cysteine. So you will get this question in number of entrance exams. Cysteine is nothing but amino acid NH2 and this side CH2 mercaptoalanine is cysteine. Mercaptothiol is here. And the 6th one is cysteine. Cysteine. And the abbreviation for cysteine is CYS and it is abbreviated as C. And when it comes to cysteine, so the thioether of cysteine or dimer of cysteine is cysteine. I can write CH NH2CO2H here and this side CH2S CH2 CH NH2 and CO2H. So the dimer of cysteine is cysteine. The abbreviation for this is cis and cis. Yes. So these are the 2 amino acids having sulfur. Next, and glutamic acid. The next letter is G. Glutamic acid. This is 7th one. Glutamic acid. Glutamic acid is nothing but, what is the glutaric acid? Glutaric acid is 6 carbon dicarboxylic acid. This is CH. Let us write this is amine. And what is the R group here? 1, 2, 3, 4, 5 and 6. So this is glutamic acid. The abbreviation for this glutamic acid is GLU and the single letter is E. Then glutamine. Glutamine. Glutamine is glutamic acid amide is glutamine. Let us write same glutamic acid CO2H CH2 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 and this is amide. Yes, amide of glutamic acid is glutamine and it is, what is the common this is GLN. The abbreviation for GLN is Q. Yes students, so 9 amino acids is covered. Then 9 under G, glycine. Glycine is the smallest amino acid which is do not have asymmetric carbon because it is attached to hydrogen. So this is alpha amino acid which do not have asymmetric carbon. Remaining all are having asymmetric carbon. This is asymmetric carbon, asymmetric carbon, asymmetric carbon, asymmetric carbons. Right. So it is abbreviated as glycine is GLY and the single letter is G. You can write it as G. Then histidine. The 10th one is histidine. Dear students, sometimes you will get minus histidine, right? So, but remembering minus or plus sign, which is optical rotation, is not important while writing the structures. So, I am avoiding confusion by removing the optical rotation. So, histidine is minus histidine, right? So, histidine is the compound having imidazole molecule. Imidazole. So this is imidazole having NH. Then this is CH2 and remaining is amino acid. So imidazole containing amino acid is histidine. So this histidine abbreviation is HIS or it can be abbreviated as H. Next comes hydroxylysine. Hydroxylysine. There is a lysine, so this is hydroxylysine. So hydroxylysine is going to have 2 asymmetric carbons. Let us write. So 6 carbon chain. So let us write first amino acid. This is amino acid. 1, 2, 3, 4, 5, 6. Right? So this side it is having amine group, which is the basic amino acid, and this is the amine which is having hydroxylysine is having hydroxy group on this carbon, and this hydroxy is downside. This hydroxy is downside, and when you see the absolute configuration, it is going to be downside, and this is hydroxylysine can be written as HYL. Next, under H, hydroxylysine. Yes. Then follows hydroxyproline. Hydroxyproline. Do you know what is the structure of proline? So this is the proline, and writing proline structure is always important, students. Proline, and you can, if you want, you can write it as like this. Hydroxyproline, and this is OH. So the absolute configuration is, this is dotted. Yes, this is dotted. This is 4s, 2s, this is 2, 1, 2, 2s, 4r. Hydroxyproline is the amino acid having hydroxy group. Right. So what is the abbreviation? This is HY P and single letter is O. hydroxyproline. Next. Yes, students, isoleucine. Isoleucine is the compound having 5 carbon chain CO2H. This is amine. 1, 2, 3, 4 and 5 carbon chain. Then this is iso position. Here it is methyl group. And this is towards downside. Methyl group, isoleucine. The abbreviation of isoleucine is ILE and it is I. This is 13, then 14 comes. 14th one is lysine. Lysine is leucine, sorry. Let me write. So this is leucine. CH3, CH2, CH, amino acid. This is LUU, L followed by 15 is lysine. This is carbon chain CH, CO2H, amine. CH2, 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 1, 2, 3, 4, 5, 6, CH2 and amine. And the lysine abbreviation is LYS and this is K. Then methionine, which is very, very important, students. Methionine. Methionine is the compound having sulfur is thiomethyl ether of alanine CH CO2H and this is amine CH2S methyl. Methionine is thiomethyl ether of alanine is methionine. So another one more carbon CH2 is there. CH2S methyl is methionine and the short name is MET. It can be abbreviated as EM. Then comes, yes, from here it is easy to write. Then it comes phenylalanine. The name itself phenylalanine says that the alanine, same alanine, write down the same alanine, then write simply phenyl group. Phenyl, that is benzene, that is nothing but benzene group, and CH2, CH, amine group, and acid. Phenylalanine, and the abbreviation is PHE. It can be F. Followed by 18 is proline. Proline is, this is the proline, NH carboxylic acid. So hydroxyproline is having hydroxy, but proline do not have this hydroxy group. Then comes 19 is serine. Serine is hydroxyalanine is serine. Hydroxy CH2 CH acid and here NH2 is serine. Serine can be written as SER and the short name is S. Proline is PROP. PROP. Proline. Yes, students. So next is 309. 309 structure is very very important, students, to remember. Is 20th one is 309. 309 is amine is downside and the hydroxy group is above side. Yes. So the way you have to write is CH3 CH. This is hydroxy group upside and this is CH amine downside and acid. This is 309. 309 abbreviation is THRT. THR and it is T. Then comes tryptophan. Tryptophan is having indole 3 acetic acid. Indole 3. So is it visible? Let me write indole. This is indole NH CH2 CH amino acid. So indole 3 acetic acid is right tryptophan and the abbreviation is TRP and it is represented as W. 309 is T and tryptophan is W. Very very important, students. And tyrosine, where can I write? Is it? Yes, I can use here. Tyrosine. 22 is tyrosine. Tyrosine is nothing but it is hydroxyphenylalanine. Phenylalanine is tyrosine. Parahydroxyphenylalanine is tyrosine. Parahydroxy. And the last one is valine. 23 is valine. Is nothing but CH. This is CH3 and CH amine and CO2H. Yes, friends. So these are all the natural occurring amino acids. Then how to remember and what are the importance? Why I wrote all these amino acids? Yes, students. There is lot of importance of writing these amino acids. The amino acids usually what is the common factor in all these things? Yes. So this is the amine group, alpha to carboxylic acid. All amino acids are alpha amino acids. Number 1. And when you see among these, some are essential amino acids and some are non-essential amino acids. What is essential amino acids? Essential amino acids are the amino acids which are not synthesized in our body. Yes, so these amino acids are not synthesized in our body. We have to take from outsource by food, by means of food. Plants will synthesize these amino acids which are essential amino acids. So the essential amino acids, when you look into the essential amino acids, asparagine is essential amino acid. Asparagine is essential amino acids. You have to take through food itself. Then follows histidine. Histidine also is an essential amino acid. Then follows isoleucine. Isoleucine essential, leucine essential, lysine essential, and methionine also is essential. Then phenylalanine also is an essential amino acid. Then comes 20, that is 309, and valine, that is 23, is essential amino acids. These essential amino acids has to be supplemented through food, right? So this is one story. And remaining are non-essential amino acid. That means these amino acids can be synthesized in our body by different pathways, by DNA and RNA. We will learn how these amino acids are going to be synthesized in our body, right? So all these amino acids are physically, they are colorless crystalline solid. That means these are white color, no color, so you can say white color crystalline solids powders, and the amino acids are high melting point solids. Yes, this is one factor. And moreover, when you look into the structures, these structures, for example, this is alanine, is having carboxylic acid group and amino group. Is it going to remain same in solution form? When you add water or when you dissolve into some solvent, it is going to ionize into, this is the ionization, CH, and this carboxylic acid going to lose proton, CO, O minus, and this amine abstract that proton to become NH3 plus. So this two ions, having two ions in one molecule, is known as amphoteric or diionic. This diionic or amphoteric ionic compound is known as Zwitter ion. It is very, very important, students. Zwitter ion. And this Zwitter ion undergoes the reactions of amine and this reactions of carboxylic acid. Yes, so it is nothing but Zwitter ion is going to be neutral compound because this amine is stabilized, this is neutralized by this carboxylic acid. Yes, since the same compound is having amine and acid, so usually amino acids are neutral. But when you look into deep into the structures, when it comes to arginine, arginine is having extra amine group. So it is basic amino acid. Arginine is basic amino acid, and this is before that, let me clarify acidic amino acids. So based on the structures, you can classify these amino acids into three categories: acidic amino acids, basic amino acids, and neutral. Yes, when you look into the structures which are having dicarboxylic acid, so usually if there is no extra carbon or no functional group, they are neutral. But when you look into the structure, this is aspartic acid is having extra carboxylic acid, is acidic amino acid. Then what else? This is also is having one. Then glutamic acid, yes, this is also having extra carboxylic acid. So this is also acidic. The name itself showing acid. Yes, aspartic acid and glutamic acid or glutamic acid. These are acidic amino acids. One more is glutamic acid. These are amino acids of having carboxylic acid extra. So these are acidic amino acids. These two are acidic amino acids. Then comes basic amino acids. Aspartic acid. Aspartic acid is lysine. When it comes to arginine is basic amino acid. This is having extra amine group. So this is basic amino acid. So basic amino acid. When it comes to lysine, yes, lysine, yes, it is having extra amine group. So it is also basic. So base, it is not neutral. It is a basic amino acid. Then histidine. Histidine is having imidazole molecule. Because of this imidazole molecule, it is also going to be basic. So base structure, basic. It is going to act as a base, right? Histidine is one of the examples of basic amino acids. Then comes remaining all are neutral. What are the important things? Sulfur containing amino acids, methionine, mercaptocystine and cysteine. Dear students, you may get a question like this. During the during the preparation of dairy products, you will get stringent smell or poisonous smell. If you pass through the dairy farm, you will get stringent smell. Why? Because milk is going to have this amino acid. This amino acid is containing sulfur. So because of this amino acid, it is going to show or have that stringent smell, which is poisonous, right? So this is how amino acids can be classified into acidic, basic and neutral amino acids. And the structural importance is, you have to remember sulfur containing amino acids and acidic amino acids and basic amino acids. Yes students, if you want you can take the screenshot of this. Yes. Next, now structure of proteins. We know that proteins are the polymers of amino acids, right? Then let us take the smallest amino acids and learn something. So let us take glycine. So dipeptide we are going to make, students. So this is glycine and this is alanine. Usually it is not going to form this peptide bond. There are different methods where you have to protect this amine group and this acid group by benzyl ester. This is by Bock, and when you take this Bock glycine and benzyl ester of alanine, and for example dicyclohexyl DCC, dicyclocarbodiamide, there are methods, but so synthetic methods is there is a lengthy chapter is there, right? So when you combine these glycine and alanine, simply it loses water. For example, I can say this is the water molecule goes out to give amide. Let me write the amide, NH2, CH2, CO, NH, CH and CH3, CO2H. So this is the amide group. What is this? Amide group. This amide group is also known as peptide bond. Peptide bond. Amide group is also known as peptide bond. Amino acids, amide group is known as peptide bond. So this is the peptide bond of two amino acids. So it is dipeptide. I can say it as dipeptide of glyala. So the common name I can write glycyl alanine, or short name I can write it as glyala. So it is a dipeptide. Proteins is a polymer of amino acids. So when I take one amino acid and do the combine or condensation with another amino acid, upon losing this water, I am going to get dipeptide or this peptide bond, which is nothing but glycyl alanine. This is known as dipeptide. For example, if I add another amino acid here to this another amino acid, I am going to get tripeptide. And further another amino acid, tetrapeptide. So further another amino acid, penta. Hexa, another amino acid, hexa. Like that it goes on increase Yes, there is a solid phase synthesis which is Merrifield amino acid protein synthesis, which you can go and add another amino acid, amino acid, amino acid. So up to 10 you can name it as peptide, decapeptide like that. But if the peptide chain is more than 10, then the compounds are known as polypeptides. More than 10 are known as polypeptides. Polypeptides. Right? So dipeptide, more than 10 peptide bonds, you can name it as polypeptide. Yes? Polypeptides. And if it is more than 100 units, it is very important, students. More than 100 units with the mass molecular weight of around 10,000 units. Don't worry about this. If the amino acid or peptide bonds are more than 100, you can up to 10 you can say di, tri, tetra. Then above 10 you can name it as polypeptide. If it is above 100 peptide units, then it is known as protein. Yes, students, are you clear now? Proteins is nothing but the peptide of amino acids. What we have seen? Number of amino acids. There are 23 amino acids. And when I combine one amino acid with another one by this way, by formation of peptide bond, if it is only two amino acids, it is dipeptide. You can name it as like glycyl alanine. If I add it is histidine, glycyl histidine, like that. And go on adding amino acid, amino acid, amino acid, you are going to get polypeptide. If the number is more than 10 amino acids, then you can say it as polypeptide. Right? So polypeptide, when it beyond or above the 100 amino acid units are there in one molecule, then it is known as protein. That means protein is a polymer of or poly units of number of units, more than 100 units of amino acids linked with peptide bond. Right? So it is the usual condition. It is the usual condition. But there is one amino acid, for example, exception I can say amino acid. There is one protein or hormone which is known as insulin. Insulin, chemically it is a protein and functionally it is an hormone. Chemically it is a protein, functionally it is a hormone, right? So insulin, which is released by beta cells of pancreas, and it is regulates the blood sugar levels. So insulin is a protein which is having 51 units of amino acids or 51 peptide bonds. So there is no hard and fast rule that all proteins must have above 100 units. When you are going to classify amino acids or polypeptides or peptides, you can consider it as 100, but there are some exceptions where insulin is one of these. Right? So 51 units peptide bonds having 51 units is known as insulin. Right? So this is how proteins are polypeptides having more than 100 units of amino acids. Clear, students? Up to here. Next. So if you do not understand here, it is very tough to understand the primary secondary structures of these proteins. Proteins are highly complex structures. Let us see the classification of proteins. So proteins can be classified into two types based on their molecular shape, on their molecular shape. Means, students, just know what we learned. So glycine, alanine, here, for example, glycine, alanine, histidine, or whatever, histidine, valine, isoleucine, leucine. You can go on construct the molecule in a linear fashion by peptide bond, and the molecule, how it is going to adjust that lengthy molecule? Is it going to circulate around it, or is it going to be like straight chain? Based on the molecular shape, these are classified into fibrous proteins and globular proteins. Yes, students. So the fibrous proteins are polypeptide chains run parallel. So I can write, for example, G, L, alanine, R, C, M, T. What we learned the short names. So M, A, L, like this. So these are going to have this linear chains. It can be this way, or parallelly it can have other chain like this, G, C, M. The abbreviations, whatever the abbreviations, E, Q. So like this, these amino acids are going to be linear straight chain, and this chain is, these compounds, fibrous compounds, are water insoluble. There is a question, students. Fibrous proteins are water insoluble. Yes. So these are like fiber-like structures. And water insoluble, these are fiber-like structures. Then what are the examples? Where do you find? You will find these fibrous proteins in hair, keratin. There is a keratin. Protein is keratin, which is present in hair. Where do you find other one? And wool-like structure. Wool, silk. Yes. So wool and silk. Silk also is animal product. Wool also is animal product, right? So keratin, wool, silk, hair. These are the compounds which are having fibrous proteins. And when it comes to other one is myosin. Myosin is a name of protein which you will find in muscles, fibrous muscles. And when you are eating some meat, the meat which is having fibrous like structures, it is nothing but myosin. So this is water insoluble. And the globular proteins. The globular proteins are usually water soluble. And these are going to have polypeptide chains coiled around to give a spherical shape. So this chain is going to coil. For example, A, L, M, C, E. So like this, it can coil, and it is going to have D. It is going to have intermolecular, or it is going to have spherical shape. So like this, it is going to have spherical globule type. It is going to have globule type like this, globule-like structure. Yes, friends. So water, these are water soluble. And for example, water soluble example are insulin. Insulin, just know what I said. Insulin is water soluble. And albumin. Where do you find this albumin? Egg albumin. Yes. When you break the egg, you will get albumin. So this egg albumin is soluble in water. So based on the solubility, I can say based on the molecular shape or based on the solubility in water. These are two types, fibrous and globular. So water insoluble are keratin. For example, if you dip your hair in water, is it going to soluble? No. So water insoluble. whereas globular proteins are spherical in shape, right, like globules. Have you ever seen homeopathic pills? Yes, balls like pills. So these globular proteins are going to be like that, and these are water soluble, for example insulin and albumin. So this is the way you can broadly divide. But is this the simple way you are going to learn proteins? No, students. There is a complex structures of proteins. So based on the structures, they are classified into primary, secondary, tertiary and quaternary structures. So this is fibrous and globular proteins classification comes under tertiary structures of proteins. Let us see one by one. So proteins, based on their structures, you can classify them into four types. Four types. Yes, the first one is primary. Primary proteins. Secondary, based on their structure. Secondary, tertiary and quaternary, right? So this is the way you can classify proteins. Then what is primary? These primary amino acids or proteins are very straightforward. So what I said, I can write like this. It is like polymer. It is like a thread combining like this balls, right? Chain like structure. So it is going to be straight chain like simple structure. So these are simple proteins. It is complex proteins or the complex structures are more complex structures. This is simple, next secondary, next end. So the structure, when you go on moving from here to there, the structure is going to be complex, and each linked with each other in a specific sequence, right? So just now what I said, if I say glycine here, lysine or methionine or whatever maybe the, if this is the case, the amino acid goes on the same one sequence. If you take one protein, if I say for example insulin is one example of protein. So that protein is not going to have randomly, the structure is specific. That means the amino acids or the amides or the peptide bonds is specific, where the amino acids are going to be arranged in a specific manner. Yes. So when it comes to secondary structure, these are secondary structures are going to be two types. Here it is the complex structure starts here itself. Yes. So two types. One is, so these are not like this straight chain. So I can write it as straight chain like this. But secondary are going to have this spherical like this helix form. So here the helix, if I say this is the helix, screw type helix, right students? So this is the helix, right side screw. So this helix is all, so the helix is nothing but this is one amino acid, this is another amino acid having different amino acids. Then why it is going to be like spherical helix? It is in helix, or the name is alpha helix. So why it is going to be like this? It is because the carboxylic acid which is there in amino acid and the amine group which is there, for example this is carboxylic acid, and the carboxylic acid which is there in this amino acid group is going to have intramolecular hydrogen bonding. The secondary structure of proteins is going to have alpha helix. So this is one amino acid. So primary structure is straight chain amides, but when it comes to here, it is going to be helix form like screw type. Why it is taking this screw type shape? Because of intramolecular hydrogen bonding. So this acid is going to have hydrogen bonding with amine of another amino acid, right? So this is the amine which is there in the molecule. It is going to form amide, this hydrogen bonding. Yes, students. So this is how it is going to have intramolecular hydrogen bonding. It is going to have this way. It is going to have hydrogen bonding. I can write hydrogen bonding like this. Yes. So because of this hydrogen bonding, it is taking this helix shape. Helix shape. Right. This is one way. Is this the only way? No. Other secondary structure, if I say beta pleated. Pleated means, so the beta carbon of one amino acid is going to, for example, CH, it is one amino acid. If I write here acid and NH, NH and acid, NH and acid, right? Like this, the complementary another. If I take this, so it is going to have, for example, amine, carbonyl, CHR, R, and here R, carbonyl, NH, R, carbonyl, NH, yes, R. So the sheets one after other there will be sheets, and these are going to have intermolecular hydrogen bonding. Intermolecular hydrogen bonding. Yes, students. So these are going to have intermolecular hydrogen bonding. This is R. Yes. Let me stick onto this. R and here and R. NH. So this is going to have intermolecular hydrogen bonding. So it is going to have intermolecular hydrogen bonding like this. So because of this pleated structures, so these are classified as sheets like pleated. So it is a complex structure, is going to have pleated intermolecular hydrogen bonding. The important point is intermolecular, here intramolecular. Yes, it is helix form. It is pleated form. It is going to have intermolecular. Yes, students. Then comes tertiary. So tertiary structures, what I said? So tertiary structure, when you look into deep, these are nothing but, just now we have learned, if it is fibrous straight chain, it is going to be fibrous. Tertiary structures are fibrous proteins or globular proteins. Globular proteins. So this is also another way of classifying these proteins, fibrous and globular proteins. Yes. So if I write in a fashion, so these are going to be like this. Yes. So it is coil type, it is further coil, self coil, right? So it is coil, globules type. See, it can be fibrous or it is globule type balls like proteins. So this is the tertiary, and the main factor of having these three structures, secondary, tertiary, fibrous, globular, alpha helix and beta pleated, what is the forcing factor which is binding these shapes? One is hydrogen bonding. Dear students, important, hydrogen bonding, or you can have sulfide, disulfide bond, yes, or I can say these are intact, intermolecular hydrogen bonding, main thing, or the attraction towards these different amino polypeptide chains or protein chains because of van der Waals or van der Waal forces, isn't it? So van der Waals forces also plays a role of binding these proteins in these shapes. Yes, students. So this shape is very important. Alpha helix shape of amino acids. Then when you take quaternary structures of proteins, quaternary are going to have this tertiary structure like this. Yes. At the same time, there will be another branch or one chain, it will have on its own, there will be chains like this. Yes, side chain or any other chain. So quaternary branched structures. So branched proteins are nothing but quaternary proteins. Quaternary proteins are complex molecules. Right, students? So if you want to learn the amino acid sequence of amino acids in any given protein, there is a way how to know the amine end and acid end proteins or amino acids. Alright. So at this level, it is better to learn up to this level. The structure of proteins can be classified into 4. Right? So primary, secondary, tertiary, quaternary. The important thing to remember is here, alpha helix structure is going to have intramolecular. Right? Is it inter or intra? Intramolecular hydrogen bonding. Beta pleated is intermolecular hydrogen bonding. And the tertiary structures are fibrous and globular. And quaternary is a branched structure. This is how, based on their structures, proteins can be classified into these 4 forms. Yes, students. So next, what I am going to say, denaturation of proteins. Denaturation of proteins. Have you ever observed when you take an egg and boil it in water, what happens? The egg albumin is a protein, solidifies. Albumin solidifies, isn't it? Albumin solidifies. So why albumin is going to solidify? Because of temperature. So the albumin is a biologically active compound or biologically active protein as long as it is undisturbed by means of temperature or by means of chemical nature or in any form. It is going to be in an active form where all amino acids, these secondary, tertiary and quaternary structures are going to be intact and they are going to act on their own biological activity, specific biological activity they will perform. But if at all, if you disturb just by temperature, the amino acids are going to, this complex structure gets solidifies or gets straightens or breaks or whatever may be. So all biological activity goes off. All biologically activity goes off in albumin. For example, if you take egg. So this process is known as denaturation. Denaturation. So natural amino acid upon temperature, if it changes or it loses its biological activity, so natural is going to become denatural or denaturation. So how to get this denaturation? By means of temperature or chemical nature, chemical, by changing the pH, just by changing the pH. Yes, if you change the pH, it is going to break or it is going to lose its amino acid, it is structures. For example, if I take milk and the bacteria, lactobacillus bacteria will produce lactic acid. Lactic acid. Lactic acid is nothing but acid, CO2H, hydroxy methyl group. So lactic acid, this lactic acid, yes, lactic acid, this lactic acid is acidic. So pH of this milk is going to be acidic, and then that becomes curd. Curd. Yes. So milk is nothing but a protein. So milk, if you drink milk, you are going to get proteins. So milk proteins are going to be disturbed or denatured by lactobacillus bacteria by releasing or converting lactic acid. So because of that lactic acid pH, it is going to become curd. Yes, students. But the question is, yes, the structure, this coil or whatever this is, it is what we are going to disturb this quaternary, tertiary, secondary. But important thing is the primary structure. This sequence, it is not going to disturb. This sequence is not going to be disturbed. Undisturbed. Primary structures are undisturbed. Remember, students. Disturbed. Primary structures are not going to disturb. So primary structures are not going to be disturbed. Undisturbed structures. By denaturation, primary structures are not going to be disturbed. So when you see these structures, when you do denaturation, you are going to get some chain. So this chemical chain, yes, there are some methods where you can go on break here one by one. So amine or acid. So by that breaking, one can come to know the structures. Yes. So globular helix get, yes. So this is how it is going to be denaturation takes place. Dear students, please subscribe my channel Venus Academy if you are not yet subscribed, and don't forget to click on like button and bell button.